Hello everybody, this is Rohan, and now we are going to discuss about the second part of food microbiology, that is regarding to the food preservations. In the last video or in the first video of food microbiology, in part 1, we have discussed about the food spoilage, what are the causes, what are the factors which cause the food spoilage, their extractors, and a lot of things we have discussed in the part 1 lectures. So in this lecture about food microbiology, we are going to deal us about the preservation aspects. सो हमें हमने तो फर्स्ट वीडियो में बता दिया कि फूड स्पॉयलेज क्यों होते हैं। अब खाना बर्बाद कर दिए तो उसको बचाने के लिए भी तो कोई तरीका होना चाहिए। क्या ऐसा तरीका है? जी हाँ, ऐसा तरीका है। आप सभी ने देखा होगा अपने घरों में कि व्हेन वी हैव टू प्रिजर्व द फूड्स फॉर लॉन्ग टाइम, वी जनरली कीप आवर cooked foods in refrigerator, or even we cook some value added products to uh, increase the storage shelf life of those processed products. So in these videos we are going to deal mainly about the food preservations. Okay, so let us start. Before that, uh, if you have not subscribed this channel yet, please do subscribe and share these videos to all other friends who is related to these courses. For example, uh, this topic now, which is food microbiology, food preservations, is not only the topic of food microbiology. This topic, when I am in university, this topic we have covered in biochemistry as well as food science nutrition, and also in post harvest technology management courses. So this is not the only topic which is only deal in food microbiology. Okay, so we will discuss everything in this video about the food preservations. So what we are going to discuss about the in this food preservation videos. So first we will see about the introductions, what is food preservations. Then we will discuss about the principles of food preservations, then how to prevent the entry of microorganisms, then inhibition methods, then destroying food preservation methods, pasteurization, method of pasteurization, then what is blanching, appertization, canning, what is low temperature, then dehydration, irradiation, fermentation, fermented foods, chemical preservation, foodborne diseases, and mycotoxins. So if you guys want this PDF notes, so you can refer or join my Telegram channel at the Rohan Agri, where you can access these notes of every videos. So stay tuned, stay connected with us. Let us learn about food preservation. So, food preservation is a process through which physical or chemical agents are used to prevent microbial spoilage of food. So, generally, we prevent the growth of microorganisms. For example, if we have cooked foods, so we keep our cooked foods in refrigerator. Why? Because the temperature of refrigerator, which is not favorable for the growth of microorganisms, that's why our food is not going to be spoiled. So these so we use some physical agents like we use high temperature, then chemical agents we have, for example we use sodium benzoate to preserve the foods. Then food preservation aims as treating food in a manner to prolong its storage life. So you can see this is the carrying products. And if we have buy some value added products in mall or markets, so if we have buy the products like pickles, okay, so for example, Kisan, we have Kisan brand, jam, jelly, marmalade. So these all are value added products. So their storage or storage life of this product is too long. So they can storage for a long period, or we can use this without any hamper of their quality and their taste. So in food preservation, efforts are made to destroy organisms in the food. So we generally uh, we do efforts to preserve the foods by destroying other microorganisms present in the foods, like if there is a presence of any spores, if there is a presence of asexual bodies of bacterial or fungi, we will kill them to prevent the growth of microorganisms, then increase the period taken by the microorganism to adapt to the food environment before they start to spoil the food. So this is introduction. So there are three general principles employed in food preservations. The first one is preventing the entry of microorganisms to food, then inhibiting the growth of microorganisms, then destroy microorganisms. So these three principles. So as we know directly, for example, what is CIDAL? What is CIDAL? There is general things, CIDAL, CIDAL, and there is static, not a statistic. This is static. Okay. So this is CIDAL. So CIDAL means, for example, we have, if you, so you have seen a lot of things like fungicide, bactericide, nematicide. So what is CID? So it is CIDAL. It is another word, cidal, which means to kill the organisms. And what is a static? A static is reduce the growth of organisms. So in cidal, for example, bactericidal, if you use bactericidal, they will destroy the microorganisms of bacteria. And if you use bacteriostatic, they will reduce the growth of microorganisms or bacteria. So that is the difference between cidal and statics. So here preventing the entry of microorganisms to food. So in these principles, we generally prevent the microorganisms. If microorganisms enter into the food, they will grow, they will sexually reproduce or asexual reproduction happen. Then there will be mass multiplication of bacteria, and there will be consumption of food, which will also spoil our food. Then inhibiting the growth of microorganisms. We can inhibit the growth of microorganisms like when we use refrigerator or when we use freezing or chilling. So during in this method, we generally inhibit the growth of microorganisms. Then we can destroy the microorganisms. How? By pasteurization method. Okay. Then what is preventions? So preventing the entry of microorganisms at sources. How we are prevent the microorganisms at sources? So when farmers are harvested the crops, then they store. So if there is a improper handling, for example, there is a lot of disease in the paddy field. For example, false smut of paddy. If there is a false smut of paddy in the field, so the spores of that paddy, false smut, will transfer to the storage also. So they will cause the disease. Okay, we will discuss in later slides. So asepsis. So what is asepsis? Asepsis is just the conditions, we make a conditions where there is no any inoculation of microorganisms, so that place is called asepsis. Then there is a good agriculture practices. So when we practice, when we handle a good agriculture practices, then we can able to prevent the entry of microorganisms. Then good manufacturing practices. So when the product is manufactured, and if there is a lot of microorganisms present in the environment, and if there is a lack of improper packaging, so we will feel difficulty, then we could not able to prevent the microorganisms. Then good hygiene practices. So that's why we have to practice these good agriculture practices, good manufacturing practices, good hygiene practices. Also always we follow asepsis. So on in this condition, we will prevent the microorganisms at their source. Okay. Then food preservation is achieved by inhibition of growth and multiplication of microorganisms by any of the following method. By reduction of water activity. So if you have not seen the first lecture of food microwaves, so where we have discussed about the relative humidity or water activity, and also the moisture content. So the moisture content is 1. If there is a moisture content is less than 1, so there will be a reduction. So when we reduce the water activity by drying and salting, by which we can preserve the food. Then reduction in pH by fermentation and addition of acids. Then we will use preservatives like sodium benzoate. Then we will use low temperatures like chilling and freezing. Then smoking, which has a drying and preservating effect, not that smoking, not snout. That is another smoking. That is the method of food preservation. Then inhibition method. So preservation of food by inhibition does not necessarily imply the destruction of organisms. So when we uh, preserve our food by inhibition method, it means not that they will destroy the all over organisms, but when we will remove the inhibition method, then there will be inoculation of microorganisms which can also spoil the food. Then spoilage microorganisms are destroyed in the food and protected against subsequent contaminations. So these are the several destroying methods you can refer here, like pasteurizations, sterilizations, cooking, canning, appartizations, which is also a part which we also call it canning, then radiation, then chemical preservations. Okay. So what is the food preservation methods? So food preservation methods like high temp, we can uh, we can preserve the food providing high temperatures. Okay, then low temperatures, because there is a favorable, any microorganisms can grow up to a 10 degrees celsius to 40 degrees celsius, but beyond 50 degrees celsius a majority of microorganisms could not able to mass multiplications. That's why we can prevent the microorganisms growth even in the low temperatures. So everything required optimum temperature for their growth and development. Okay, for example, if we are human beings, so we can maintain our body temperature up to 5 degrees celsius. So by maintaining, if your outer temperature is 5 degrees celsius, we will use sweater, we will wear sweater. And if the temperature is beyond 40 degrees celsius, we will use cotton clothes. So every microorganisms or living beings has their optimum uh, temperature required for their growth and development, even in the microorganisms. So low temperature, chilling and freezing. So microorganisms growth inhibit. Then dehydration. So we will use some salts material which will reduce the water activity. Then reducing the water activity. So reducing the water activity like salting, pickling or adding sugar. So when we will use when we will use sugar and salt, so there will be change in the water activity which can stabilize the food for a better shelf life. Then we have irradiations. Irradiation is also called as we will use radiations to preserve the foods. Then we have fermentations, like fermentation methods like we have curd, then we have idli. Okay, then we have wine, beer. So a lot of alcoholic products which will generally uh, produced by the process of fermentation methods. Then we have chemical preservations. Then let us discuss about the pasteurizations. So he is the great, very milestone scientist, that is Louis Pasteur, who was a French scientist. So during industrializations, so the French, vodka, wine. So there is a lot of cultivation of grapes in France. So people generally consume French wine, not French wine, grape wine. So grape wine was not preserved. So pasteurizations, which is this is the technique developed by the Louis Pasteur. So because of their name, we use the pasteurizations. Then in so first time pasteurization method was used in the preservation of grape wine, because the demand of grape wine in the French is more. Why? Because European country has a winter condition climatic there. So winter there is always low temperatures. So people will use or people will consume wine to maintain their body temperature. So we should generally use the wine to maintain our body temperature. But we don't know about in other countries. We will use as a for stuffs. So it is the process of heat treatment at a specific temperature and times. So during pasteurizations, so there is a definite temperatures and there is a definite times. So we will use. So that is the on that time the food will uh, change their properties. Okay, and all the microorganisms also destroyed by these processes. So in the market, if you have seen the pasteurized milk is there, then pasteurized juice is there. So a lot of products in the market. So pasteurized is aimed at destroying all pathogenic microorganisms without affecting the nutritive value of the food, like milk, fruit juices, non-alcoholic beverages, and wine like apple wine, cider, and grape wine. Okay, then changes during spoilage. So three methods of pasteurization is there. So low temperature long time, high temperature short time, flash method, or ultra high temperature. So this is the common question asked in many examination. What is LT and what is their conditions? Then ST, then what is ultra high temperature? So low temperature long time, where we provide 63 degrees Celsius temperature or heat for 30 minutes for those products. So all the microorganisms will destroys. So this is LT process of pasteurization. Then we have high temperature short time. So high time, so on the temperature of 72 degrees Celsius at the time of 15 seconds, so the products will go through in this uh, process. So high temperature short time, this is the another method of pasteurizations. Then we have ultra high temperature, where we use the temperature of 130 degrees Celsius for 1 to 2 seconds. So these are the pasteurized products if you have seen in the market. For example, in Bihar or Jharkhand, so there is a dairy product Sudha. So they have introduced a product of pasteurized milk which you can store for a 30 days. So that is a innovation which is going by the process of these types of methods. Then exposed to steam or hot water. So we generally, if you have seen, if you involved in kitchen, so if you have seen there is a products like potato, then if you have cooking meat product, so we generally first we expose into the steam. Okay, to why we are exposing in a hot water? So we are exposing in hot water to kill the pathogens. It will kill the pathogens, and it does not any affect the food color. Then evacuates the air. Then it inactivates the enzymes. Then mostly used during canning. So during exposure of steam or hot water, so even if canned product is there, we kept all those canned products in the hot water. Okay, then what is canning? What is appertization? So intermittent heating and cooling. So in these appertization methods, we generally we use the products for a heat the products for a longer time and for a longer cooling. So in this method, that is appertization, which is developed by the Nicholas Appert. So because of their name, the term is appertizations. So he is also called as a father of canning, and first time he used appertization methods in their home. Then what is boiling? Everybody knows what is boiling and cooking. So at 100 degrees Celsius we boil the products. Then long term preservation of food. So we have some steps we should follow for the long term preservation of food. Like we should clean the products, then we can cut the products, then we can blanch the products by bottling, then sterilization, then storage. So this is the steps for the long term preservation of food. So can you tell us some examples of this type of preservation foods? For example, have you seen kurkure, then chips, lays? So these all are the products. For example, potato chips. So they we can see in the mall and in the shops of grocery. It is the storage life of those kurkures and chips are very long. Even we can store for 1 to 2 months. So how it is going to be possible? So first of all, their processed products, so even they provide some nitrogen in the pockets. For example, if you buy the kurkure products for 15 rupees, so it will be like full of air. It was like it was big pocket. But when you open the uh, kurkure packets, so there is nothing, there is a less amounts of kurkure is there. So this is the methods of even the marketing, and this is the methods of preserve the foods. So even nitrogen gas is filled in those products, in those packets. So what is low temperatures? So two methods are employed to arrest microbial growth and multiplications. The first one is chilling, which is cold storage, and the second one is freezing. So in chilling during, we maintain the temperature of 0 to 15 degrees Celsius, and the common chilling temperature ranges between 4 to 5 degrees Celsius. So all the fruits, vegetables, milk and products are in this type of chilling temperature ranges. We preserve these all products in this type of temperature ranges. Then in freezing is keeping food at temperatures between 0 degrees Celsius to minus 35 degrees Celsius, like meat and meat products and other several fruits. Then what is the effect of low temperatures? So low temperatures are used to retard chemical reactions and actions of food enzymes and to slow down or stop the growth and activity of microorganisms in the food. A low enough temperature will prevent growth of any microorganisms, as we have seen in freezing. In refrigerator we generally see. So spores are not usually injured at all by freezing. However, most parasites are killed by freezing. So the some spores of fungi and bacteria are not killed by, but there are a lot of parasites which present in the products which can killed by the freezing. Then we have drying and smoking. So what is drying? So drying is a natural sun drying. So we generally uh, spread the products in the presence of sunlight. So that's how. For example, if in the villages, like if you have seen the mushroom drying process, so mushroom drying process or a lot of in traditional mushroom dyeing process, the person who does not, who is growing in the home, and if they have not a proper dryer, so what they do, they simply spread their mushroom in the presence of sunlight. Okay, or by mechanical dryer, like, so we have a lot of mechanical dryer where we preserve the food like dry fish and the milk. So dry fish in the state of Kerala, this is the common methods to preserve the dry fish. Even they preserve the fish for a long time. Okay, that is dry fish. Then even the milk is also stored. Milk powder, not milk. So milk powder, if you can see the Amul milk powder. So the spoilage of that product is not, because if they are the products of non-perishable communities, even in the instant. And then smoking, so we provide some smokes, drying plus wood smoking like fish and meat. So this is the smoking, not like that smoking. Okay, so drying plus wood smoking provide, so we can able to preserve the fish and the meat products. So by lowering water or osmotic pressure, we can preserve the foods. How? So addition of high content of salt, like sodium chloride and sometimes nitrates and nitrites. By addition of high sugar during pickling, we provide some salt, and even we lower the pH during braining. So we addition some salt solutions during braining. Then use of radiations which are lethal to microorganisms like ultraviolet rays and the alpha rays. No, sorry, it's gamma rays. Alpha, beta, gamma rays. Most commonly used ultraviolet rays, water, utensils, fruits and vegetables. Then gamma rays also used in the packed foods, meat, poultry and egg products. Then we use cold sterilization. So what is cold sterilization? Generally the cold sterilization is generally the radiation provide to the products. So that is the cold sterilization. Cold sterilizations. So the it is the method to preserve the food by radiation. That is cold sterilizations. Then radiorizations, pasteurization using radiations. So that is, so RAD for radiations, and pasteurization. So urization is for, so that's how this word is radiorizations is formed. Then microbial mediated transformation of food. So this is the fermentations. So in fermentation, the production of alcohol and acids happen. So during the fermentations, the alcohols and acids adds, we use alcohols even in the acid produced during fermentation used in the preservation of the products. We adds new flavor and taste during fermentation process, which improves the nutritional quality, like milk, uh, the conversion of curds by the milk, then butter, cheese, yogurts. Then we have cereals, like conversion of cereals in beer, then idli, tempeh, soya sauce, etc. Like by the fruits like apple wine, grape wine, even in the vinegar. Okay. Then vegetables, pickle, sauerkraut. Sauerkraut is the products of cabbage. Then meat, sausages. So these are the products which are preserved by the methods of fermentations. Then chemical preservations. So use of antimicrobial chemicals to kill or inhibit the growth of microorganisms. Okay, so there are several classes of antimicrobial chemicals. Like in class first, we use salt, sugar, vinegar, honey, or oil. Okay. Then in class seconds we have benzoates, sorbates, nitrates, sulfides, and a lots of antibiotics is there. So benzoates. So we use benzoates to preserve the jams, jelly, juices, and vegetables. Then sorbates. We use sorbates to preserve the cheese, olives, and a lots of subproducts. Then we use nitrates to preserve the meat. Then we use sulfides to preserve the wine and dry fruits. Then we have foodborne diseases also. So when we consume the food and if that was spoiled, so we are sick. We will get sick. Why? Because the presence of microorganisms there. Okay, and a lot of pathogens even in the present in the spoilage food. So when we consume that pathogen enter in our body, they multiply and they produce some toxic chemicals which which harm our body. Okay, and which will produce a lot of diseases and create a lot of dysentery, a lot of things is there. So food-borne diseases are acute illness associated with the recent consumption of food. The food involved is usually contaminated with a disease pathogen or toxicants. Such food contains enough pathogens or toxicant necessary to make a person sick. Okay, then foodborne infections. So these are two types of foodborne diseases: the foodborne infections and the foodborne intoxicants. So let us discuss first foodborne infections. So in the foodborne infections are illness caused by the entrance of pathogenic microorganisms into the body through contaminated food and the reaction of the body tissues to their presence. Okay, so these can either be fungal, bacterial, or viral or parasitic foods. Borne infections tend to have long incubation periods and are usually characterized by fever. So presence of live microorganisms is must to cause the infections. So if there is a not presence of live microorganisms, how they can affect our body? They can't. Then bacterial foodborne infections. So there are two types of foodborne infections, bacterial as well as viral foodborne infections. So the foodborne infections is bacteria like cholera, so which cause the disease cholera, then salmonellosis, then typhoid fever, okay, then shigellosis. In shigellosis, there is a it is associated with a dysentery, stomachache, gastrointestinal problem. Then Escherichia coli. So we have discussed Escherichia coli in the aquatic microbiology. So what is this is the if there is a presence of Escherichia coli in any products, it means there is a presence of fecal matter. So this Escherichia coli is the indicator of fecal contaminations. Then infections, Escherichia coli infection, Campylobacteriosis, then Vibrio para, hemolyticus, and Listeriosis. Then viral foodborne infections include hepatitis A, Norwalk virus, and poliomyelitis virus. So Norwalk virus is related to the gastrointestines. It create a lot of problems in the gastrointestine. Now this is the table for the major foodborne infectious diseases. So you can see here the diseases, their causal organisms, their incubation period and characters, then major food involved. So you can see and read these things by pausing the videos, or you can take the screenshots, or you can download even the notes. So foodborne intoxicants which lead to the food poisonings. So this is the common disease if you have seen if you are living in the boys hostel, or if you are living in the quota, or even in the if you are making their own food. So a lot of boys or a lot of girls in the hostel, they are affected by food poisoning. Why? They are not serving us a good quality of foods. Okay, and if they have consumed spoiled foods, even they are the causes of food poisoning. So in India, there is a lot of boys and girls are affected by food poisoning. So illness caused by ingestion of toxins produced by microorganisms present in food. So if we not provide proper quality and proper hygiene food, so there will be problem of food poisoning. So when food poisoning, there is a stomach ache, headache, and there will be dysentery problem. So this is the very disgusting diseases. So microorganisms need not present in food. Its products is sufficient to cause illness, have short incubation periods, and are characterized by lack of fever. Foodborne intoxicants can be classified into bacterial intoxicants, then fungal intoxicants, chemical intoxicants, plant toxicants, then poisonous animals. Okay, so bacterial foodborne intoxicants. So like Staphylococcus aureus, it causes the disease. Then Bacillus cereus, Clostridium perfringens, then Clostridium botulinum, which cause the botulism. So they all are produce some toxic chemicals which cause the disease in the human beings. Then these are caused by the consumption of metabolites produced by fungi when growing in food, like aflatoxins. So aflatoxins are released by the Aspergillus flatus, Aspergillus niger, and Aspergillus penicillium. So aflatoxins cause the mycotoxins. You can see a mycotoxins. So mycos means mycologist, mycorrhiza. So we generally refer fungi for myco, and toxins is with chemicals which are harmful for the human beings or animals. So amanitin, Amanita phalloides, then phalloidin, Amanita phalloides, then gyrometrin, Gyromitra species, then Orcra toxins, Aspergillus ocraceus. Then the fermented foods. So fermentation, what is fermentation? So microbial mediated conversion of sugars into alcohol acids, metabolic process of microorganisms to derive energy in the absence of oxygens. In the absence of oxygens. Then it is fermentations. So fermentation of food by age-old process known to mankind like bread, curd, idli, wine, etc. Fermentation in food. Mode of preservation adds flavor, taste, new food conserved and enhance bioavailability of nutrients. Then fermentation of food. Alcoholic fermentations. Sugars to alcohol and carbon dioxide mediated by yeast, wine, beer, shake, bread. Then there is two types of fermentation foods. So top fermenting is like Saccharum cerevisiae. So Saccharum cerevisiae which use this is the common yeast we use uh, during preparation of wine and ale. Then we have bottom fermenting is like Saccharum carbo calburgensis. So this is the common yeast used during the production of beer. Then fermentation of foods, acid fermentations. So sugars to acids mediated by the bacteria and fungi like yogurt, cheese, sauerkraut, pickle, idli, and sausages. Then we have lactic acid fermentations. So lactic acid fermentations two types, homo and heterolactic. So homo means identical or hetero means different. So homolactic. So sugars to only produce lactic acids like Lactobacillus plantarum, Streptococcus, and heterolactic, so sugars which produce lactic acid as well as produce alcohol, carbon dioxide, and a lot of other acids. This is the fermented food groups and their examples like dairy products, meat products, cereal products, fruits and vegetable products, legume products, fish products, beverages and crop products. Then we have miscellaneous products of fermented food. Then some fermented foods products like wine, which are the material required for making wine, which is fruits, and what is the microorganism? Saccharomyces cerevisiae. Then beer, so cereal grains, the material required. Then Saccharum carbonagensis. then yogurt, which is produced by the milk, then bread, which is produced by the flour, then milk, acidulous milk, then distilled beverages, then sausages, which is produced by the beef or pork. That is some food products and their predominant organisms, then their starting materials. So thank you for this. Uh, so this is the complete of food preservation. So if you have any questions, if you have any doubts, please do write in the comments. And if you have liked this video, please do subscribe the channel and even like the videos. So, and please share the channel because this channel is new and nobody knows about these channels. If you share these channels, so a lot of students will be come together and we will learn a lot of things. Okay, so thank you. Stay tuned us and stay connected with us, guys. Okay.